Hello! In this tutorial, I will be explaining the required folder hierarchy and contents of input data for a LAM analysis. The folder hierarchy is a crucial part of the functioning of the tool, as it is used to gather information about the samples and sample groups in addition to keeping the data separate. First, it should be noted that the quality of the analysis is highly dependent on the quality of the input data. This means that the microscopy images should always be masked to only contain intensities of the samples and to make sure that object detection and segmentation have as high accuracy as possible. For object detection, we have used Imaris and its spot detection algorithm to segment nuclei from midgut images. However, other object detection algorithms can be used as well, as long as you can provide object coordinates for LAM. Following object detection, the obtained data must be organized in order to use LAM. First, each sample's data must be placed within the analysis directory in separate folders. The analysis directory should only contain files created by LAM and the user-given samples. Any additional files may cause errors when using the tool. The folders must be named according to practice, where each sample has its sample group at the beginning, followed by the sample's name separated by underscore. Within each sample folder, different object sets are placed in separate folders. These should be named with the sample group and sample name as before, but extended with object set's name and the text of your choice, both separated by underscores. These dataset folders are called channels, named after microscopy channels, but can contain, for example, datasets of co-localized objects. Each folder will be joined with similarly named datasets from other samples, which will then be analyzed as a unique dataset. We have named the data folders according to the labels or markers that the objects originate from, followed by the extension statistics that is added by Imaris. All data pertaining to the channel must be within its folder and in CSV formats. The only required file that must always be found is position.csv that contains the coordinates of the objects. The data in this folder is exported from an Imaris analysis, and many of the files are not used in the analysis. All data can also be placed within the position.csv in wide formats, but this requires altering of the default settings that are designed with Imaris exports in mind. Within the position.csv, the XYZ coordinate columns must be named position X, position Y, and position Z. The data must also contain column ID that has unique number identifiers for each object of the channel. These IDs must match between data files for correct merging. All other columns in the CSV are left unused unless specifically named in LAM's settings. LAM only accepts numerical or empty values within the data columns. All samples must be structured similarly to the one shown. However, all samples are not required to have all channels. LAM will only add the data into channel's full dataset if it is found, and the missing data file will not cause errors. All samples can also be provided with a data file containing an anchoring point, alternatively called a measurement point, with abbreviation MP. The MP is used to align each sample in the full data matrix to reduce compounding error caused by proportional variation between intestines. If no MP is provided, each sample is joined to the matrix as is. However, it is nevertheless recommended to give the MP data to maximize biological correspondence of regions within the data matrix. Due to the variation in midgut length, the further you go from the anchoring point, the greater the error in cross-sample comparison. The user can also use the split and combine method to minimize this error. See the split and combine video for further explanation. The MP is provided by creating a channel folder with the name MP as per default settings. This folder should contain a position.csv with only one object that has its x and y coordinates. This object should point at a biologically distinguishable segment, such as the midpoint of the R3 region of the midgut. This concludes the input data tutorial. In the next video, I will go through how to use LAM to create the vectors required for data projection, or alternatively, how to input coordinates of self-created vectors.